All right, so let me start sharing my screen so you can actually see the presentation. Okay. As you guys know, they are what you eat. And first, what I want to do is start off with like a little bit of a disclaimer. Okay, so parasites are really cool organisms, but some of these photos might be considered graphic. So for example, I have a photo of fish dissection. So I apologize if you just ate lunch, but yeah, just a heads up, fair warning. You can't say I didn't warn you, but anyway, on with the show. So as Mr. Seifert already mentioned, I graduated from Weiser back in 2013, so it's been about 6 years now. And while I was at Weiser, I was super involved in the ag department. So not only FFA, but just the ag department itself. We did not have SRI yet, but what we did have was a bunch of cool ag classes. So in 10th grade, you do a current topics in ag project. So it's a semester-long career project. And I actually did mine in parasitology. And then I think the following year, I went on and participated in the public speaking competition, where I also did a speech about parasites. So this passion for parasites and this interest in parasites has been with me for about 8 years now. So after high school, I graduated, and I went on to Kutztown University. And there I participated in multiple research projects. They range from looking at bird boxes to electrofishing for fish in the local creeks. And we dissected those fish and looked for parasites in them as well. So now, as Mr. Seifert said, I'm a graduate student at Ball State University out in Muncie, Indiana. And I'm going to talk to you today about a general overview of parasites, as well as go into what I'm actually researching. So I went a bit of a different career path where I went straight to my PhD from undergrad. So like I said, I want to give you a little bit of an overview of what I'm going to be doing the next 5 years in graduate school. But first, let's talk about generally what parasites are. So the question is, what is a parasite? So the Center for Disease Control defines a parasite as an organism that lives on or in a host, which is another organism, and gets its food from or at the expense of a host. And in ecology, we like to call this a plus-minus relationship, where the plus is one organism benefits, and the minus is when one organism is harmed. So of course that means the parasite is the one benefiting, whereas the host is the one being harmed. So there are two major groups of parasites, endo and ecto. So do you guys know what endo means? I guess you're not old enough to take Latin for the root yet. But endo means within, and ecto means outside. So within the body versus outside the body. So commonly known ectoparasites. So the first one I want to go over is called the black-legged deer tick, or the deer tick. So this is one I'm sure you're all familiar with, because living in Pennsylvania, basically every single tick carries Lyme disease. So ticks are becoming a lot more prevalent because their seasons are becoming longer due to climate change, and like we said, they help spread Lyme disease. So can you guys name another common ectoparasite? I think you guys talked about them yesterday. There was a guy there.
Who came to talk yesterday?
No. All right. Well, it's a mosquito. So, as you guys, I think maybe some of you attended his talk, but they're responsible for transmitting diseases such as malaria, West Nile, and Zika. So they also will suck your blood. So these guys are considered parasites because they get their nutrients from our blood. So major types of endoparasites. So like I said, remember, endo means within. So these are the parasites that are found inside of a body. So the first one is called a trematode, also known as a fluke. So these guys are cool. So you can see in the picture that there are two arrows, and the one arrow is pointing to what's called an oral sucker, so towards the end of the parasite. So these trematodes will actually feed on your blood by this oral sucker. And then towards the middle of the parasite, the second arrow is a ventral sucker. It's a little hard to see in this picture. But that ventral sucker is used to help stick to the walls of like the inside of your intestine or the stomach. Another common endoparasite is called a cestode, so a tapeworm. So these guys have what's called a scolex, which is the head region. And on this scolex, you can see the backwards facing hooks at the top. So those will help it latch on to like your intestine. And then they also have 4 oral suckers also on that scolex region. What's really cool though about tapeworms is they don't actually have a mouth. Instead, their bodies are super flat, so all nutrients are absorbed through their external body covering. And last but not least, so another common endoparasite is called a nematode or a roundworm. As their name suggests, they're more round, so to speak, but they're also really cool because they differ from trematodes and cestodes in that they have a complete digestive system, just like us. So, you know, food goes in one way, comes out the other for us. So the same goes with roundworms. So they have a complete digestive system, and they'll like live within your intestine. So sometimes parasites have different types of life cycles. So sometimes they'll have a direct life cycle. So this means there's only one host involved in which the parasite can reproduce as well as is considered effective. So here we have a prime example of how the bird can shed the parasite eggs into the environment, and then from its own eating, it can become like infected by these eggs. So one type of host, so one bird. However, sometimes parasites can have multiple hosts. So we call this a complex life cycle. So here in this picture, you can see that the first intermediate host is the snail, and then the second one is going to be an amphibian, so the frog. At this point in their lives, the parasite are not able to reproduce, they're simply just growing. But once the parasite is able to get to what's called its definitive host, so the bird, it's able to reproduce and produce more eggs, and then the life cycle just continues and continues. So, I want to get into more crazier parasites, probably what you guys were more of expecting. So, first one I want to go over is called daughter. So, did you know plants can also actually be parasitic? So daughter is the orange plant that you see in the picture, and they have a modified root system called haustoria. So these haustoria will actually enter the stem of another plant and then start sucking the nutrients out of that plant instead of, just having its own root system within the ground. So this one's cool. This is actually a grain-eating amoeba. I remember in high school there was something about it in the news, but it's mainly prevalent in warmer areas. So Texas and Florida are the areas that have the highest cases in the United States. But the trophozoite stage is the part that's infected. So the grain-eating amoeba must enter through your nose from contaminated water, and then it will travel to your brain where it can cause a severe brain infection, which ultimately leads to death. So it's super fun. So another one, so you'll notice that this is a fish, but it doesn't have a tongue. Instead, it has a tongue-eating louse, which is a giant isopod. And a more common example of an isopod is the roly-poly or the pill bugs that you can find in like your garden. So what actually happens is this louse will enter the fish through the gills, and then the female will actually attach herself to the tongue. sever all the blood vessels to the tongue so that the tongue will fall off, and then she'll substitute herself as the fish's tongue, and the male will stay in the gills to mate with the female whenever she's ready. So, have you heard of the crazy cat lady? Because there might be an explanation for that, and it could be a parasite. So Toxoplasma is a protozoan parasite that actually infects cats. So this parasite also has a complex life cycle. So its first host is a rodent, actually, or a bird. And this parasite is cool because it will manipulate the host to change its behavior. So a bird or a rodent, when it becomes infected with Toxoplasma, they'll actually change their behavior to be more showy so that they'll actually get the attention of the cat, which is its definitive host. So, for example, the rodent, let's say, will come out of its hiding hole and run around so that the cat will actually find it and chase it and grab it. So then, from the cat, it can actually be transmitted to humans. So we can get and become infected with Toxoplasma by eating raw or undercooked meat, or by simply changing litter boxes. So we can actually just inhale the infective stage of a parasite, which is another reason why they say pregnant women should not change a cat's litter box, because they could become infected. And when a female who is pregnant becomes infected, they can actually pass the disease onto the fetus. And when you're infected with Toxoplasma, you can exhibit flu-like symptoms, blindness, confusion, hence crazy cat lady, headaches, and seizures. Last one. So this is true. I can't make this up. He ate a slug on a dare, became paralyzed, and died. This happened recently in Australia because this kid ate a slug that was actually infected with what's called a rat lungworm. So this is a parasitic nematode. So you can actually see in the picture with the nematodes that they're a complete digestive system. So they're pretty transparent underneath the right settings. And rat lungworm uses a snail or a slug as an intermediate host, and then an animal with a backbone. Typically rodents, but that includes us since we have a backbone as its definitive host. So, one way, obviously, you can get this parasite is by eating food contaminated with the larval worms of the parasite. So, if you ever eat escargot, which is snails or slugs, make sure that they're fully cooked. Otherwise, it could affect your brain and spinal cord and cause temporary paralysis and obviously, ultimately, lead to death. This is very grim. I apologize. But anyway, so the question is, who can actually be hosts? Well, here we see a picture of a deer, and in this deer, they can also carry parasites. So in the picture, that is a roundworm that I found in the coop of deer last summer. Snails, as we have seen time and time again, they can also serve as a host. So this snail is infected with a type of roundworm that also causes behavioral changes. So the snail will go out to the edge of a leaf and start waving around the actual worm inside of it. So those are like kind of where the antennae would be, so that it will get eaten by a bird, and then it can carry out its life cycle. Fish can also be hosts to parasites. So in this picture, we're seeing what's called thorny-headed worms. So it's a type of roundworm that I found inside of the intestine of a smallmouth bass. So also make sure you cook your fish. As we saw earlier, plants can also be parasitized. So this is called a goldenrod gallfly, and she'll lay her eggs into the goldenrod gall plant, and it'll form this like tumor or growth on the stem, and inside you can actually find the larval form of her. And apparently they're great for fishing. And of course, humans. We can be host to parasites as well. So overall, we really only see parasites in the context of them being bad. You know, we hear them in the news all the time in terms of spreading diseases and whatnot. So are there actually any ecological benefits of parasites? And before I move on, I want to ask what ecology is. I want to define this. So ecology is the study of the interactions between abiotic and biotic factors. So if I showed you these two pictures, can you tell me which is the abiotic and which is the biotic factor? Which rock is abiotic? Yeah, Lydia? What? You guys speak up. So Dwayne "The Rock" Johnson is biotic, right? Yeah, so he's living, whereas abiotic is not living. So The Rock on the left is not living, obviously. So you guys seem to understand that. So I'm going to move on to how parasites can help shape communities. So a community in ecology is a bunch of different species living together and interacting with their environment within a habitat. So let's say we have an infected rabbit. So because the rabbit is infected by a parasite, this can decrease its fitness, where fitness is defined as the ability for an organism to survive and reproduce. But if instead the rabbit becomes healthy, that increases its fitness, so the ability to grow and reproduce. So when rabbits are healthy, they produce a lot of offspring. So we go from one rabbit in the population to having 6 more. And when we look at this in the grand scheme of things, so looking at it from a trophic pyramid standpoint, if there are more rabbits in the lower part of the pyramid, that means that there is more energy available to the foxes in the upper part of the trophic pyramid, which then could increase the fox population. And in ecology, we call this bottom of effects. So bottom of effects means something happening at the lower part of the food chain affects the upper part of the food chain. So for this example, increasing the prey population can therefore increase the predator population. Another way parasites are beneficial or can affect ecosystems is that they can actually act as corridors. So corridors simply meaning links between, say, dry land and an aquatic habitat, so like a stream, lake, or a creek. And they can alter these trophic interactions. So let's say we have a grasshopper, and let's say that grasshopper is infected. And I want you guys to keep in mind that infected also means that they represent energy within the ecosystem. So if this grasshopper is infected and is being manipulated by the parasite, it will actually jump from dry land into the water where it doesn't belong, where it can then be eaten by a fish. And then that fish itself can become infected. So from this, we have a transfer of energy that wasn't supposed to be in the aquatic habitat, and what we get, that means that there's just more energy to go around within the ecosystem. So one more. So parasites can increase connectedness within an ecosystem. So one statement that I always read in the paper is that parasites are ubiquitous. And ubiquitous just means that parasites are literally found everywhere. And they represent approximately 40% of described species, yet we don't really see them in our textbooks on food webs or food chains. So I'm going to show you a picture, and it's going to look pretty complicated, but this is just a food web of different organisms, and you can already see that there are many links within this food web of an ecosystem. But look what happens when we add parasites. So when we add parasites, it looks completely different, and you can see that the ecosystem becomes a lot more complicated and has so many more different links. So when you actually increase the links or connectedness, this actually means that the ecosystem itself is more stable, in which stability of an ecosystem means the ecosystem is more resistant to change. So essentially it means that it's a healthier ecosystem because if some perturbation, so an invasive species comes or something, the environment itself won't change. However, this is still kind of a pretty controversial hot topic in the parasite realm in terms of they're still not sure whether or not we should include parasites in food webs or food chains. But by simply looking at this, it looks like we're missing a lot of the big picture. So before I tell you about my project, does anyone have any questions about parasites in general? We're going to switch gears a little bit. All right, seeing no hands, so I'm going to take that as a no. So my project is called Does Land Use Affect the Host Diversity Begets Parasite Diversity Hypothesis? Or relationship, sorry. Wow, don't even know my own dissertation. That's good. So, biodiversity. So first I want to define biodiversity in that it's a bunch of species living together within a habitat. So here we see the white-tailed deer, wild turkey, a groundhog, all living together in North Park. So when you increase biodiversity, it's said to increase the resource availability within that ecosystem. So that just means the food, shelter, water. It also increases resiliency. So that is also being resistant to change. And then increasing biodiversity can also lead to increases in productivity. So productivity just means the amount of biomass, so how much plants are there, how much energy is within that ecosystem. With increases in biodiversity, we also see a decrease of to the risk of local extinctions. So that's kind of going off that resiliency idea where if something were to happen, it's not likely that a species would go extinct within that ecosystem if some sort of negative factor came into the ecosystem. And increases in biodiversity gives way to what's called the dilution effect. And the dilution effect is simply the more different types of species you have within an ecosystem, the less likely disease is going to be transmitted. And a possible reason for this is, for the example of parasites, is that having more species may make it harder for them to get to their target host. So when we think about biodiversity, we think about that charismatic megafauna, so those lions and tigers and bears. But when we look closer, like we said earlier, we don't really think about parasites. And there's a lot of research going on with parasites versus in their relationship with their hosts. And what that has led to is called the host diversity begets parasite diversity hypothesis. So what that means is they're finding a positive relationship between host richness and parasite richness. So the more different types of hosts, the more different types of parasites should be within that environment. So currently there are many anthropogenic impacts on biodiversity. So we are, as humans, negatively impacting biodiversity. And there are 5 main reasons. The first is through the introduction of invasive species. So this is a New Zealand mud snail. As the name suggests, they are from New Zealand and not from the United States. So we introduce those into our creeks and they can outcompete our native snails, so for food, shelter, and whatnot. Overpopulation, which then of course can give way to pollution, which we all know can alter turbidity of creeks, so how clear the creeks are. They can affect the animals itself depending on what we are polluting them with. So, for example, pills and whatnot that we get from the pharmacy, when we flush those, say in the sink, down the toilet, whatever, dogs are actually showing mutations from these different types of pharmaceuticals. Another is overexploitation. So a classic example is the cod industry. So we thought that there were infinite amount of cod within the ocean, but so we just kept fishing and fishing and fishing, but then the population eventually collapsed. So it's taking more than what is actually there. And last but not least, destruction of habitat. And I want to talk about habitat loss today, and specifically in the context of changing the land use. So for example, that's going from a forested area, like old growth forest, to making it a field of corn, which is super popular in Pennsylvania, or changing the forest to, say, a college campus, a high school, a town, whatever, just as long as it's being developed. So tying those two ideas of the host diversity begets parasite diversity relationship and changing the land use, I want to go over the objectives of my study. So I want to look at previously published literature and analyze it to see if changes in land use affects that host-parasite diversity relationship. So that's not collecting my own data quite yet. It's looking in already published studies to see if that relationship already exists. And then I want to actually collect my own data. So my second objective is to determine the role of land use in shaping the relationship between stream fish diversity and parasite diversity. And last but not least, I want to then switch gears and determine the role of land use in shaping the relationship between terrestrial snail diversity and parasite diversity. So these are my hypotheses. So we have, once again, host richness and parasite richness on our axes, and thinking that forested is going to be the one that is quote-unquote most accurate in terms of representing a natural environment between the relationship between host and parasite richness. I'm predicting that they'll have the most positive relationship in showing that hypothesis. And then I'm thinking with agriculture and developed, that positive relationship between host and parasite richness is going to decrease a bit, just because there are so many different anthropogenic or human impacts associated with agriculture and developed land. So the question becomes, how do we actually categorize land use? So what we can do is we can zoom in to, say, the watershed that we're looking at, and then we can take those land uses that we have defined, so develop forested agriculture, and isolate them within our watershed, and then, so this is what we're looking at, we can determine that for this one, agriculture is the most abundant based on that count. So we have a little bit of data for the meta-analysis, but overall it's not that conclusive. So like I said, this is from other studies, so this is not my own data, but agriculture is represented by that golden brown, and we're actually seeing that positive relationship between host and parasites, whereas for forested we're not, which is the exact opposite of my hypothesis, but that's science for you. But it should be noted that these sample sizes are not representative because for agriculture, we had 6 sample sites, and for forested, we only have a sample size of 13. So nothing can really be drawn from this. So that leads me...
Jenna, can I interrupt you real quick?
Yeah.
So if you looked at streams running through like farmland, snails there had more parasites than streams running through forests. Is that what you're saying?
Yes.
Oh.
Yeah, exactly.
Cool. And that's opposite of what you expected.
No, that is definitely not what I expected.
Okay. All right, thanks.
Yeah, no problem. So that leads me to now collecting my own data. And what's crazy about science that I'm sure you guys haven't thought of is you need permission to do science, and it costs $10. So I have to apply for this collecting permit because I'm sampling animals with a backbone, which is something I didn't know going into this, but to apply you need to do a proposal and then you give them a check for $10. So it does cost money to do science, if you didn't know. So how am I actually getting my samples? So for fish sampling, we're doing electrofishing. So I mentioned this before, but just in case you guys don't know what it is, it's using an electric current to attract fish, and then you just scoop them up with a net. Another way we're doing this is called seining. So it's using a giant net and basically scraping it across the bottom, lifting it up, and hoping there's fish in there. So then we bring the fish back to the lab and we dissect them. And I think from what Mr. Seifert has said, is you guys will have the opportunity to do this very soon. So we dissect them, and when you're dissecting the fish for parasites, you have to separate the organs out into different petri dishes so that you know the parasites came from that organ specifically. And then, hopefully, you find a parasite. So this is a nematode I found in the intestine of one of the fishes. For snail sampling, there's two ways we're approaching this as well. One is you can literally just go out and look for snails using your eyeballs for like an hour. The only issue with this sampling technique is that it can create bias. So when you use this technique, an issue is that you typically find way more snails that are bigger than snails that are smaller because eyeballs. You know, our eyes can only see so small. Then I have this state-of-the-art sampling technique of using wet cardboard and putting it on the ground and leaving there for a day and then coming back. And when you lift up the cardboard, hopefully there's lots of snails underneath it. And then we bring them back to the lab and perform live dissections on them, because when the snail, if you kill them, they'll actually just start to disintegrate, which will destroy your parasites, and that just would not be helpful in any way, shape, or form. So the question is, now what? So this is, as Seifert said, my first year in grad school, so I actually don't really have any data to go over or anything or any results. So I'm currently processing fish and whatnot. But if you were to get anything out of this presentation, it's that passions can really start young. I entered Kaiser High School in 2009, and in 2010, I found my passion for parasites, and now I'm 567 miles away working on my PhD on things that I love. So, as students of SRI, I greatly encourage you to go out, do some awesome science, and follow your passions. And that's all I have.
All right, thanks, Jenna.
Yeah.
Anyone have any questions?
Lisa.
That Lisa?
Yeah.
Like what made you interested in parasites?
So here, let me try to stop sharing. So when I was in 10th grade, I was super interested actually in the parasitic disease, just because they were absolutely gnarly. You know, the parasite that goes into your eyeball, and then, I don't know, have you guys seen Monsters Inside Me, that Discovery Channel show? But then I realized halfway through that I really don't want to work with, like, patients and people. So then I switched gears to more an ecology side.
Jenna, do you have a favorite parasite?
A favorite parasite? I would have to say, it's called a schistosome. It's a type of fluke, and it can cause the disease schistosomiasis, because that is the parasite that got me interested in parasitology. So they are they're a fluke, so they have that oral and ventral sucker, and they can go into humans and cause really bad diseases. But that's the one that got me interested in parasites, so that's probably my favorite. Otherwise, I would say a pork tapeworm. Don't eat undercooked food, guys.
Jenna, do you eat raw sushi?
I do, but sushi grade fish is different than like the fish you get actually at the grocery store to like cook for dinner. So they're a little different. It's quote unquote safer. I take my chances.
Me too. I do have one other question, but I want to see if anyone else has a question first before we let you go.
I actually have a question. I don't know. I have a question because I watched that Monsters Inside Me show. It's fun. Don't you get worried? And I came in a little into your presentation. Aren't you worried about contracting a parasite or anything like protections? What kind of protections do you have in place for yourself and your other research?
So this is actually super funny. So when I first started researching parasites, so my mom and my dad, so you guys know her, Mrs. Lewis, they said the one rule was I'm not allowed to contract a parasite. I guess no food in the lab. I wear gloves whenever I dissect them, but I would say that the protection is fairly minimal, but I'm not eating or anything. All the parasites I work for are not like that toxoplasma where I could inhale like any type of parasite or anything. It all would have to be ingested. So number one rule in lab, don't eat.
Thank you.
Yeah.
You got another question? Yep. Go ahead, Ariana.
Do you know what the African guinea worm is?
Yeah. What's it called? I haven't done a lot of research on that because I'm not very medical, but I have heard of it. Do you have a fun fact to back on it for me?
What? No, not really.
That's fair.
What?
Is it pretty nasty?
Yeah, definitely.
Yeah, they cause crazy symptoms and diseases. That I do know.
All right, Jenna, so my question is, you're talking about ecology and like how complicated parasites are and how they're interacting with tons of other species. So you're doing your PhD, and your project sounds pretty complicated, but if you're sitting here and you're in 6th grade or 8th grade or 10th grade, like, I guess how would you recommend, if anyone's interested in parasites, like how they start, like what kind of easy questions are there that need to be answered about parasites still?
Honestly, parasitology is such a new field. You can ask any question, really, and anything you do will be contributing tremendously to the field. It could be as simple as, how does this parasite get into its host, or what happens if I raise the temperature of the water for an aquatic snail? Will the parasite die as well? So it can be simple questions like those. Just going out. Actually, this is a really super easy, fun project. If you go out to a creek and you just collect aquatic snails, put them in a tank, and if they're like infected, you can actually see larval forms of parasite emerge from the snails, like within 24 hours. It's really cool.
So we've already done 2 stream studies during SRI this year, so I don't know if we get back out to the creeks or any of those, maybe we can look for some snails or something.
What?
I just said we've already done some stream studies, so they might have some experience getting into the creeks and finding snails and stuff already.
Okay, yeah, I recommend doing that. You can literally see them within 24 hours. It's crazy.
All right, got another question here.
What electives in high school did you take?
What?
Did you take any electives in high school to prepare you for I took essentially
classes you could. I remember that. AP Environmental Science is probably the best class you can take or at least I'm very biased. What's it called? You do a lot of fun projects, and you really learn how you impact the world. Spoiler alert, you do, or you might not do this anymore, but you do like a trash audit sort of thing, where you monitor how much trash you produce. But that class really made me passionate about environmental science. So my PhD is actually in environmental science, and I got my bachelor's degree in environmental science. So I would say that class I highly recommend, as well as Mr. Seifert's biotechnology is really good. And then, I don't know if you guys still do this, experiments in agriculture. Do you guys still have that class?
Yeah, so we have a couple of research courses now, but experiments in ag is still running too.
Yeah, that was a good class. I was in that class by myself, you guys. But no, that was a good class. You can develop your entire own research project, and it really does get you excited about doing research. You have to form your own hypothesis, come up with your own research question. So it's really good in developing those skills as well. And then, of course, 9th grade agriculture, you can't go wrong. It really sets you up for success in life.
It's like we're paying you to do this, Jenna. Thanks.
I know, right? Man, where's my sponsorship?
All right, any other questions for Jenna? Anyone interested in, a little more interested in parasites now? Is anyone afraid to go like outside now?
What do you
All right, so Jenna, thank you so much for spending time with us. If you don't mind, I'll leave Zoom open for just a couple of minutes, and if any students didn't want to come up or like ask questions in the whole group, they could come up and ask individually. Would that be okay?
Yeah, it's perfectly fine.
Okay. All right, so let's give Jenna one more round of applause. And,